I'm I'm making two videos which will overview the playlists I have on prokaryotic cells and bacteria. This is the second of the two. The first focused on prokaryotic cells in general, their variation, and then archaea. This one's going to get more into life cycles, what is used for energy and for a carbon source, and focus more on the eubacteria. And so one of the keys in this playlist is just the variation. So for example, clearly bacteria can move, all right, because some of them have flagella. Some of them have these pili, which allow them to, adhere to cell surfaces. So there are variations, and these variations are significant. Sometimes in a health setting, the idea that bacteria can move is very important in infection. Bacterial flagella, as mentioned in the previous video, is different. They are different from the flagella which we find in eukaryotic cells or in archaea. They can allow different kinds of swimming. All right, and that bacteria don't have to be limited to one single flagella. They can have multiple kinds of flagella. Here's another type of movement known as corkscrew movement, which is allowed here. And so the next video just mentions that some bacteria have a single flagella, some have multiple flagella. They can be at opposite ends of the cell. And thus, if you're looking at difference, trying to identify bacteria or looking at the variations, then clearly there are differences between different bacteria with these flagella. Okay. One of the main aspects of microbiology is understanding just how bacteria make a living. And if you're going to make a living, there are a bunch of things that you need. You need energy, you need carbon, you need nitrogen. And so how do bacteria do this? Well, it varies, and it's important then to discuss these variations to understand bacteria. But then also it's important to understand life on Earth. And so how does one get energy? Well, energy can be gotten in a lot of different ways. So for example, if bacteria are exposed to minerals, so for example, in geothermal water, if you ever visit a hot spring, they may display the minerals which are available. In deep sea sediments, there are minerals. Well, bacteria can be found here, and they can convert certain minerals from this reduced form to this oxidized form, and in the process they can get energy. All right, so some bacteria are lithotrophs. They oxidize inorganic reduced molecules in order to get energy from it. So how can one get energy? Well, one can oxidize just inorganic materials and live on minerals as an energy source. I'll define these terms in a second, but they can then vary in how they get their carbon. So there are both lithoautotrophs and lithoheterotrophs. All right, so that is one way to get energy. A second way to get energy is to use sunlight and perform photosynthesis, in which case a bacteria is what is called a phototroph. There are different kinds of bacterial phototrophs. And so what I'd like to do, just because bacterial variations are so immense, just kind of define some terms here. And then as I go through different bacterial groups, further define them, with specific examples, which I think will be more useful. So here is a bacterium. It's called a blue-green alga or cyanobacterium. It performs photosynthesis. So does rhodospirillum. They perform photosynthesis. So they are both phototrophs in that they're getting their energy from the sun. Now, as I'll pursue later, they vary. One's using chlorophyll and is green and is releasing oxygen gas as a waste. One is using bacterial chlorophyll and is producing sulfur instead as its waste, because hydrogen sulfide is its electron donor as opposed to water. And so they both perform photosynthesis, but they're using different pigments, hence the different colors. One's releasing oxygen as a waste, one is not. And so sunlight can be used as an energy source. So can chemicals like organic molecules like sugar. So for example, here's E. coli, which would be a bacteria living in our large intestine. How do they live? Well, they're using some molecules of our diets so that it's chemicals such as sugars, proteins, lipids, etc., which is where they get their energy. Now, I get my energy by breaking down sugar and protein. I'm using chemicals as an energy source. As I convert, say, sugar into carbon dioxide and water, energy is released. And some bacteria do the same. So they are chemotrophs. And so here I've just gone through 3 different ways that bacteria can get energy. They can be lithotrophs, which oxidize inorganic minerals. They can be chemotrophs, which are breaking down organic molecules for energy. And then they can be phototrophs. Okay. Now, a second very important question is, where do you get your carbon? Because I am a carbon-based thing. This is carbon molecules. These are sugars and proteins and nucleic acids and lipids. So where do we get our carbon? Well, it turns out there are variations in where you can get your carbon. And so we can even see that here in 2 different kinds of purple synthetic bacteria. All right. So if you were to ask rhodospirillum, you're a carbon-based thing. All life is carbon-based. So where did you get your carbon? And the answer is from molecules such as sugar. So me, I have to eat things, all right? We animals, we live off of other things. We have to eat sugars and lipids and proteins from other things. That's where we get our carbon. And if you get your carbon by eating from other living things, then you are what's known as a heterotroph. And so this bacteria may be using sunlight for energy, but it's using organic molecules like sugars for its carbon source. The black atoms here represent carbon. And so it is a heterotroph. But plants, cyanobacteria, which we'll get to in a second, and then this purple photosynthetic bacterium, they use carbon dioxide. So sunlight is used to fix carbon. It's called carbon fixation. So carbon dioxide plus water, if you add energy, you can rearrange that to make sugar. So you don't need to ingest sugar from another living thing. You can make your own. And if you can do that, if you can use inorganic carbon, say carbon dioxide, to make sugars and proteins, you are what is known as an autotroph. So this bacteria is a photoautotroph because it's using sunlight for energy, and it's using carbon dioxide as its carbon source, and it can convert inorganic carbon into organic carbon. Whereas rhodospirillum is a photoheterotroph. It can get energy from sunlight, but it cannot use carbon dioxide as its carbon source. It has to then ingest it from other living things. Now, once again, I'm just going to mention words and then get through them as different examples pop up. There are different ways of being a chemoheterotroph. So bacteria who get their energy from organic molecules, and who are getting these organic molecules from other living things. How does one do that? Well, you could live in dead stuff. You could live in, dead material. You could live in rotting leaves in the soil, and you could decompose dead stuff. And so then it's the organic matter from dead things that is both your source of energy and your source of carbon if you're a chemoheterotroph. You could be a parasite living in another thing and causing disease. So it is your host, which is the source of the organic molecules, providing both your energy and your carbon source. So some chemoheterotrophs are parasites. Some are actually predators. As we'll see, there are some bacteria which eat other bacteria or eat eukaryotic cells. And so they, just as I am a predator, all right, whether it be bananas and lettuce or pigs and chickens that I eat, I am getting my energy and molecules from, my energy and carbon molecules from other things. Some bacteria are predators, getting their energy and carbon molecules from other things. So chemoheterotrophs get both their energy and carbon from the organic molecules of others, but there are different ways of doing that. You can be a decomposer of dead stuff, you can be a parasite on a living host, or you could kill the other organism to get it, in which case you are a predator. Now, one last thing that keeps coming up in subsequent videos is, where do you get your nitrogen? Do you need your nitrogen? You sure do need your nitrogen, because here's DNA. And if you remember, DNA has nitrogenous bases. These are the pink atoms. So you don't have DNA unless you have nitrogen. All of the nucleotides, like adenine, thymine, etc., it relies on those nitrogen atoms. You don't make protein without nitrogen. See these amino acids? All amino acids that make up proteins have an amino group, or more than one, and each amino group has a nitrogen atom. So as living things, if we have no nitrogen, we can't make DNA, we can't make protein, and we die. Okay, so nitrogen is something that living things need. Where do we get it? Well, you'd say, look at this air. It's 79% nitrogen gas. Nitrogen gas is the most abundant part of air. That's easy. Why don't I just get my nitrogen from air? Well, the fact is you can't, because this is a triple covalent bond. It is such a strong bond that even though most of the air that you are breathing is nitrogen gas, you don't have the ability to convert nitrogen gas into a usable form of nitrogen, like say ammonia or nitrate or nitrite, which can then be incorporated into biomolecules. You can't do it. No animal can. No plant can. But bacteria can. And so here, once again, just visiting a theme from the previous video, why do we study bacteria? Well, some people study bacteria because they alone are interesting and important, etc. But let's say I don't care about bacteria. Well, you need to study them anyway, right? Because you would not have nitrogen in your DNA or nitrogen in your proteins. In other words, you wouldn't have DNA or protein and you would not be here if it wasn't for the fact that there are bacteria which can take nitrogen gas out of the air and incorporate it into a usable form, like ammonium nitrate or nitrate, which then can get incorporated into biomolecules. So only prokaryotes, not all of them, some of them, only prokaryotes can perform what's called nitrogen fixation, converting nitrogen gas here on the left into something like ammonia. And then some other organism can convert ammonia into nitrate or nitrite. All right, this can be done by some free-living bacteria. I give you some examples. Some free-living cyanobacteria. I give you some examples. And some symbiotic bacteria. In a little bit, I'll show a video of a plant root where there's bacteria living inside the plant cells. Why would plants want bacteria living inside their plant cells? Well, if these bacteria fix nitrogen, that means this plant can live in nitrogen-poor soils. It can live in a soil where it would otherwise die, right? And so very often other organisms will provide teamwork, and say, hey, bacteria, I'll feed you if you do something then for me, like give me nitrogen in a usable form. If you were to study microbiology, one of the important concepts which comes up is the cycles of the molecules on Earth, like carbon, for example. So there are things that can take carbon out of the air. The autotrophs, like plants, green algae, which are eukaryotes, and many bacteria, they can take carbon out of the air, make it into biomolecules. The biomolecules work through food chains, things die, and the carbon then goes back to the air. There is a cycle. And bacteria are very important members of the cycle as decomposers, as photoautotrophs, you know, et cetera. Well, there is a nitrogen cycle. So nitrogen comes out of the air. It's converted into ammonia. It can be converted into nitrates and nitrites by different bacteria. I give some here. Then it can then get incorporated into living things in their food chains, becoming DNA molecules, protein molecules, etc. Decomposers then return nitrogen to the air as nitrogen gas. So there's a nitrogen cycle. Bacteria are essential in this nitrogen cycle. There are other cycles, which I'm not going to get into. There's a sulfur cycle. Living things need some sulfur, and that cycles from going from inorganic sources to organic sources. So once again, who cares about bacteria? Well, we do, all right, because the stuff of living things, whether it be the carbon, the nitrogen, the sulfur, you know, et cetera, we, like, for example, we animals, we get it from other living things as parts of food chains, right? And very often bacteria are essential in these cycles. Now, as I start going through, you know, different examples, a very important group of bacteria to talk about are the blue-green algae or cyanobacteria. So a lot of people don't think of them as being bacteria because algae, you know, is their nickname. But most algae are eukaryotic cells. So, you know, cyanobacteria is perhaps better. So they are not eukaryotic cells like green algae or red algae. And they are their own separate kingdom of bacteria. Okay, and so they are, my fault. They're their own separate phylum in the kingdom Eubacteria. So there's a kingdom Archaea, there's a kingdom Eubacteria, and just like there's you know, a phylum Arthropoda in the animals and a phylum Mollusca for mollusks in the animals, there's different phyla of bacteria, and different phyla perform photosynthesis. So notice that here's one of those purple photosynthetic bacteria. Cyanobacteria are different. Now, cyanobacteria are arguably among the most important organisms on the planet, especially when one considers the fossil record. If you could take Earth's history, all right, Earth was formed 4.6 billion years ago. You could essentially split it in half, all right? For the first half, there wasn't any appreciable oxygen in the atmosphere. In the second half, there was, although the oxygen levels slowly increased. It isn't like it went from like none to a lot. You know, it went from none to gradually more and more and more to, you know, modern standards. And so if Earth went through this drastic change about, you know, 2.3 billion years ago, what caused this change to take our planet from, you know, anaerobic, where, you know, life had to live without oxygen, to aerobic, where life could now depend on oxygen because it was present? And the answer is right at this point, the blue-green algae, the cyanobacteria, evolved. They had a new type of photosynthesis, which could put oxygen in the air as a waste. So cyanobacteria changed planet Earth. And notice it is after this point that the first eukaryotic cells evolve, which need oxygen, all right? And so eukaryotes, which require oxygen, virtually all of them, and then the animals, which require oxygen, they evolve in the aftermath of the world that the cyanobacteria have helped to make. They made the planet different, which would have caused the extinction of lots of bacteria. Oxygen would have been the first form of air pollution. There are lots of bacteria today, lots of bacteria living in your intestine, for example, which die in the presence of oxygen. They need an anaerobic environment. Oxygen is toxic to them. And so the cyanobacteria, which put oxygen gas into the air, dramatically changed our planet. So, you know, once again, it's a form of bacteria, and some people, you know, perhaps don't care as much about bacteria, but the world would be completely different without bacteria. What these cyanobacteria evolved the ability to do was to take carbon dioxide and water and using sunlight to make it into sugar. So they are using sunlight So they are phototrophs. But because they can use this carbon from an inorganic source and convert it into glucose, they are photoautotrophs. So they can use inorganic forms of carbon to make their biomolecules. Now, in the process, they produce oxygen as a waste. Now, once again, there were other photosynthetic forms of bacteria, which were probably older than them from stromatolite fossils that we have. But this was a new type of photosynthesis. And once again, the oxygen that it provided as a waste changed the planet. When we look at them under the microscope, a couple of things jump out. They're bigger than typical bacterial cells. They're more complex with their infolding of their cell membrane. But also, look, they often live in colonies, which is unusual. Most bacteria are just, solitary. And so this is a level of complexity that lots of bacteria don't have. Also, if you look along their strands, every now and then you find a cell which has decided to be different. All right, it has specialized, it has differentiated. Now, not the way that your bodies do, where, say, some of your cells have become nerve cells and some of your cells have been muscle cells, not to that level. But nevertheless, more so than we see in any other prokaryotic cell, where some cells are deciding, we're living in a group, I'm going to be different though. Some of these different cells can then decide to fix nitrogen, which benefits the colony. So sometimes this is a nitrogen fixing structure. Sometimes it's a type of spore, which would then be resistant to environmental change. So here you can see different kinds of cyanobacteria. Now, cyanobacteria can actually then live inside animal cells. Some sponges have cyanobacteria inside of them. So the whole sponge, even though it's an animal, it's more or less performing photosynthesis and getting some of its energy from the sun. And we can see that in other organisms as well. So cyanobacteria, once again, they're important for lots of things. They can actually be symbionts living inside others. But even if they're not, then the sponges are filtering seawater and they eat the blue-green algae. So where do animals get their nitrogen? Well, very often they get it from what they eat. So sponges can ingest blue-green algae directly. A big fish like a shark eats little fish, and the little fish might eat little arthropods. And the little arthropods might then be taking this out of the seawater. So the nitrogen, which is being fixed in blue-green algae, often ends up being the nitrogen in animals, because animals can ingest free-living cyanobacteria if they're filter feeding water, or there are cases where cyanobacteria and other bacteria can live inside other living things as symbionts, and survive that way. So here we see cyanobacteria. This one's kind of neat because every now and then you can see, some movement as the cells kind of fold. They can have a variety of shapes. They can, live in colonies. They can be solitary. They can have, this more spirochete shape. So there's great diversity. Remember, it's a phylum. All right, so it's not a species or two. It's a phylum of species, incredibly diverse. And I'm sorry. Let me get back to the symbionts. So not only can you find them by themselves, but you can also find them in lichens. So a lichen is a symbiotic relationship between a photosynthetic organism, often cyanobacteria, and a fungus. All right, so here are a symbiotic relationship where fungi and cyanobacteria are living together. You can actually find cyanobacteria living inside some sponges, and then corals as well, although the primary endosymbiont of corals is a eukaryotic alga. You can have blue-green algae there as well. And maybe, you're not that impressed, all right, because maybe you think, okay, so cyanobacteria, they do photosynthesis, they're green, they release oxygen as a waste. That's not that impressive, because don't plants do that? And the answer is, well, plants do, but there's a reason, as we'll pick up in the next topic of eukaryotes. Plants have the descendants of blue-green algae, of cyanobacteria, living inside them. All right, so if you look at a plant cell, it's not all green. It's got these green blobs in it. Now, I'm sure you studied in earlier classes that those are chloroplasts. Yes, but chloroplasts are the size of cyanobacteria. They're the shape of cyanobacteria. They have their own chromosomes, like the chromosomes of cyanobacteria. There are free-living cyanobacteria like prochloron, which are the relatives of these. And so the way that plants and green algae got to be green doing photosynthesis, releasing oxygen as a waste, is that living inside them, they have cyanobacteria, which are green, performing photosynthesis and releasing oxygen as a waste. So cyanobacteria truly have changed the world. We would not have green algae and plants if it weren't for the descendants of the cyanobacteria, which are living actually inside their cells as chloroplasts. So these are chloroplasts, but the chloroplasts are descended from the free-living cyanobacteria. Once again, here we have different kinds of them, and the fixing of nitrogen that they do is critical. All right. So cyanobacteria are incredibly important. And also, I mean, if you were going to list the types of organisms which have had such an impact on our planet that they've actually changed history, well, then the cyanobacteria would certainly be in that list. And once again, I just have this other video stressing that there are actually different kinds of photosynthesis performed in bacteria. So cyanobacteria, they invented a new one releasing oxygen as a gas, but there are other ones as well as in these purple photosynthetic bacteria. So the previous part on cyanobacteria was concerning the photoautotrophs. Getting their energy and their carbon, their energy from the sun, and then their carbon from inorganic sources. I had said that among the chemoheterotrophs, there are different ways. Some bacteria are actually predators. So when we consider the microbial world, there's actually predators here. So some bacteria secrete enzymes, which then diffuse away from the cells, which kills other cells. All right, and then as these cells die, their proteins, their lipids, etc., are now in the environment, and then this bacteria, Lysobacter, can now absorb those molecules, using them both for energy and as a carbon source. So this is one type of way that bacteria can prey on other organisms. Myxococcus does the same. It can cause cells to burst and absorb nutrients. So that is one type of predation. There are others as well. Now, some bacteria can live inside other cells. If this is in a multicellular organism, then we often refer to this as a parasite. But when they do it to a unicellular organism, it'll end up with the death of the cell. They kill it. And so then we would refer to this as a type of predation. And so here you see a bacterial predator, which was able to attach to its prey, penetrate the cell wall, and then inside the cell make use of its molecules for both energy and as a source of carbon. So it is a type of bacterial predator. There are others, and some of these others can, in addition to preying on bacteria, can actually prey on eukaryotic cells. So while typical food chains involve eubacterial cells, which then prey on bacteria, it can also go in the reverse direction as well, where some bacteria are capable of preying on eukaryotic cells. Here are a few other examples where a bacterium, such as this vampirococcus, kind of being like a vampire, can then approach another bacterial cell, perforate the cell wall, and then absorb the organic molecules for both energy and as a carbon source. So this example is of vampirococcus, which is now attacking another bacterium, Promatium. But in the next example, there is Vampirovibrio. Vibrio is the name we give of, say, a comma-shaped bacteria. It is actually attaching to a green alga, and thus is a predator on a eukaryotic cell. Okay. So there are, once again, different bacterial lifestyles, and chemoheterotrophs can be decomposers, getting their nutrition and carbon molecules from dead stuff, and thus very important in food chains because they recycle nutrients. They are very important. Some are parasites, and some are actually predators. Okay. I have a lot of videos here that touch on various things. Bacteria can obviously change over time. Natural selection can act on them, and so if we were modeling just evolution and evolutionary changes, so I have a video here that's just showing how with the use of an antibiotic that a population can change, and antibiotic resistance can become more and more common. So this is why we shouldn't overuse antibiotic in our population, because we will then cause more bacteria to be able to resist that antibiotic, and antibiotic resistant strains of microbes are a serious health threat. I have a couple of videos which try to simulate a field trip if you were studying bacteria and sampling different parts of the environment, just as if you were looking for birds or frogs. You would say that, hey, there's a diverse number of species, and each one kind of specializes, that certain ones are found only in certain habitats. So in the deep forest, you'll find these species, while in the wetland, you'll find these species. Well, the same thing here. Whereas if you were to sample for, say, methanogens, you would find them, say, in the mud, but not, say, in the plants or in the open water. And you'd find only some species, so only the pink and the aqua ones, for example. But if you were to go to the floor of the ocean, you wouldn't find any. But if you were to go into the ocean mud underneath, you would find more. If you were to go to deep vents, you would find more. In fact, you would find some species here which only live there, like Methanothermococcus. There are some that you would find in intestines. And so just like if you were an ecologist studying any other group, you would say, oh, there's a diversity of species, and some live in very specific environments. Some biologists study bacteria and come to the same conclusion. And then I have a subsequent video that does something similar with the blue-green algae. So once again, how do some biologists study bacteria? Well, for some, it is a question of medicine, while in others, they're studying more the ecological aspects of microbes. So once again, if you were studying the cyanobacteria, you would find different ones in different environments. And so, that is of interest. Because they're aerobic, you wouldn't find any in anaerobic environments like intestines. And so once again, there would be different ones in the ocean, different ones in actually living inside sponges for example. And so just a few other videos, as I had mentioned before, it is so important to be able to fix nitrogen that some organisms like plants set up symbiotic relationships with bacteria which can fix nitrogen. And so here you're going to see the cells of a plant root, a legume, like beans. And you'll see that inside the roots, the roots include these nodules, and inside the nodules are, so these are cells of the plant root, but you see lots of little dots here. Each dot is a bacteria. So this plant is feeding bacteria living inside its cells, but in exchange, the bacteria are fixing nitrogen, which the plant needs. And this allows these legumes to grow in nitrogen-poor soils where other organisms, other plants would not be able to grow well. Just then kind of wrapping up this topic with some other concepts that biologists might need to know. While no bacteria have true sex, where two haploid cells with one copy of each chromosome then fuse to each other to make a diploid cell with two copies of each chromosome. There are several types of genetic exchange known in bacteria, and if, say, one were to then go into genetics, these can be important. So some bacteria can take up DNA from the environment, often from dead cells, and then instead of digesting it, they can actually then perform recombination so that the red DNA from some dead organism is actually inserted then into the host chromosome. This is what's known as transformation, and bacteria can get a new trait as a result. So if they take up a gene from the environment, this gene could then allow them to make a new protein, which they otherwise would not have been able to make. And so that is a form of genetic exchange, even though it's not sexual reproduction. Bacteria can get new genes that way. A second way that bacteria can get new genes is through what's called transduction, where a virus, which normally infects a bacterial cell and causes it to burst, which will kill the cell, sometimes a virus mistakenly incorporates yellow bacterial genes into its capsid rather than the white viral genes. If this viral particle then infects another cell, it would then inject yellow DNA from the first bacteria into the second cell and introduce genes from one bacteria into another. Once again, that is a form of genetic exchange known as transduction, which uses viruses as an intermediary. Some bacteria actually do approach each other and exchange DNA. Now, it's not sexual reproduction where two haploid sets of chromosomes then fuse. And typically it involves this extra chromosomal loop of DNA known as a plasmid. All right, but here is a cell, all right, which can then approach another cell. Their cell walls can fuse, and DNA from the first cell, this loop called a plasmid, can then pass into the second cell. And so, once again, while bacteria do not undergo true sexual reproduction, they can have several forms of a genetic exchange, such as transformation, where they can take up DNA from a dead organism or a lice cell, transduction, which involves in virus, or conjugation, which is the exchange between two living cells. Now, this occurs in nature. It's important in the spread, say, of antibiotic resistance, which is important for those who practice medicine. But it's also important in genetic engineering. So, for example, if bacteria can take up DNA They could actually take up DNA from a human source, such as a human gene that biologists are trying to incorporate into a bacteria. Say if you take the gene for insulin and insert it into a bacteria, you now have a bacteria which can make human insulin, which is far better than going to a slaughterhouse and trying to get bacteria from, insulin from an animal to treat diabetics. And so a growing field of biotechnology is inserting genes into bacteria so that bacteria essentially become living factories to make a molecule that, say, a hospital wants to treat their patients, or that the industry wants for some reason. So transgenic bacteria are increasingly important. Other bacteria can be engineered so that they can, say, digest petroleum after, say, an oil spill, or absorb sulfur to minimize the effect of, say, acid rain or the pollution of sulfur in the area around coal mining. And so there are a number of applications there. I had mentioned extremophiles that can live in extreme environments, and I have a number of videos that go into them in far greater detail. Even have a song in here as well. And so what these videos go through, and once again, the purpose of this video I'm making now is just to provide an overview to kind of point students at these other videos, that they could pursue further. But it goes through, well, how does one get to be a cell that lives at 80 degrees or 100 degrees and become a thermophile? Now, some cells don't like that temperature, but they can survive it. So there's a difference between, say, being thermotolerant, where you can survive a high temperature, but you would prefer normal temperatures, versus a thermophile, which needs high temperatures. You can only find them at high temperatures. So once again, with extreme environments, some organisms can tolerate it, but some actually like it. So phile means to love, a thermophile or a hyperthermophile. These are organisms that need high temperatures. And once again, as I mentioned before, there are changes that one can make to, say, the cell membrane lipids. And so if you have your lipids branch a bit more, if you have them exist as a monolayer, that allows cells to work at a high temperature. You can change the amino acid order of your proteins so that the proteins aren't denatured by high temperatures. The PCR, which is so important in taking a small sample of DNA and making a lot of it. So for example, at a crime scene, if a little bit of DNA is found, we can amplify it to make a whole lot of DNA. That uses enzymes coming from thermophile bacteria, because now you can essentially heat your sample up to the point where the double-stranded DNA molecule separates, but that the enzymes don't denature because they came from heat-loving bacteria. So that was a key in developing PCR, to take enzymes from bacteria which were thermophiles. Now, some bacteria, they can tolerate cold temperatures, and then some are psychrophiles, which actually prefer cold temperatures below, or at freezing. And so you can find them in glaciers, you can find them at the bottom of the oceans. And if you were to ask, then what does one do? Well, once again, there can be modifications of the protein structure, which then prevent the cold temperatures from changing the structure of the protein, and to ensure proper folding even at cold temperatures. Some extremophiles are called halophiles because they can survive very salty conditions. Very often, adding salt is a way of killing things, because in salty conditions, water tends to leave cells and the cells dehydrate. But some cells have adapted where they can live in that, and some actually prefer these environments. And so once again, you might ask, well, how can cells do this? Well, you can then have different scenarios where some halophiles do what's called salting in, because if the water wants to leave the cell because the environment is saltier than saltier than inside the cell. One of the things that cells can do is bring, say, potassium chloride into the cells. And because there's already a high concentration of salt inside the cells, then the osmotic pressure is balanced and water tends not to move. So some halophiles do what's called salting in, while others perform what's called salting out. They keep the salt outside their cells by accumulating things like sugars, amino acids, etc., inside the cells. So there can also then be pumps which can remove ions which diffuse in. And one of the advantages of living in a salty environment is if you live in this high sodium, sodium could come in and you could make it do a little work for you as it comes in. Now, there are cells at the ocean floor where the pressure is greater. There are bacteria which can live even deeper at the bottom of the ocean, and there are bacteria which can live in rock deep underneath the Earth's surface, where the pressures are simply enormous. Once again, they have adaptations which allow them to do that. So they can change the amino acids of their proteins, change the fatty acids of their cell membranes, and then other differences. So for example, here are the 20 amino acids, but what is found is that if you were going to live in an extreme environment, some of these might help you do that. And so as you look at thermophiles adapted to heat, or psychrophiles adapted to cold, or barophiles adapted to pressure, there might be an excess of certain amino acids as an adaptation for that environment. Barophile is, I think, the term that I got used to as far as bacteria which live at great pressures, but technically barophile means weight. And so the term which is now being used more is piezophile, which I think is the actual word for pressure. Some extremophiles, they live at a high acid concentration. And so they have to deal with temperatures or changes in pH. Some actually have adapted so that all of their proteins will not misfold in the presence of acid. So they actually have adapted to being acidic, which is unusual. Most of them then adapt to acid conditions by pumping the acid out. So there's different ways of adapting to high acid environments. Before I go to the next one, very often an extremophile doesn't have to be one or the other, because if you live at the bottom of the ocean, it's not only a great pressure, but it's also very cold. If you live in the rock underneath the Earth, it's not only great pressure, but it's also very hot. Lots of these extreme environments are also acidic. All right, so we not only think that early life was hypothermophiles that adapted to a high temperature, we also think that they were acidophiles living in an acidic environment. So once again, we consider these to be odd environments, to be weird, but actually they might have been the first environments, and environments which are actually very common today. So much so that more than half of bacteria might live in these extreme environments. So they're really not that extreme as a result. Some environments are alkaline or basic, and there are microbes which adapt to those. I have a little song if that helps on extremophiles. And so once again, just a very quick overview. But we underestimate the great quantity and diversity of life which lives in these extreme environments. There's no one here, I live where it's hot. I'm not in my views, I'm not singing the blues. So that's the thermophile, so that's the extremophile song, if you're interested. Finally, I just want to point out that I have a playlist that goes through more lab techniques. A lot of biologists who are studying bacteria have to identify the bacteria or count the bacteria. So how does one do that? How does one identify the bacteria in a patient? Once again, I'm not going to go through it now. This is more of a lab thing. And so, those who need this can go through this playlist. But for example, I'll go through the steps of a gram stain so that one could identify bacteria as being, gram positive or gram negative, or to determine whether or not the bacteria makes spores, so that a lot of bacteria they form this resistant structure which can survive the winter or survive harsh conditions. Now that's important in environments, but it's also important in medicine, because if you're sterilizing medical equipment to prevent infection, if the bacteria in question can make spores, then that changes everything about how much, how hot it has to be and how the material has to be sterilized. And spores, because they are so resistant, so for example, like anthrax spores, from what I've read, there were islands north of Great Britain where anthrax spores were spread, which are still uninhabitable. So, as a form of a bio weapon or as a concern for doctors, the presence of spores are important. Lots of bacteria vary in what they can metabolize. So some can metabolize starch. All right, now not only would this affect where they could live, but it's also a way that you could identify the bacteria. You could put it on a plate and see if the bacteria in question can metabolize starch. A lot of bacteria die in the presence of reactive forms of oxygen, like hydrogen peroxide. But then others have an enzyme called catalase, which can produce oxygen gas, which then bubbles away. And so you can test for that by taking your bacterial sample, adding hydrogen peroxide, and then looking for bubbles. So not only would this, say something about the bacteria and where it might be able to live, but also you could help identify it. So if you have a bacteria, say from a throat culture, and you're looking at what it might be, these bacteria are catalase positive and they would bubble. These are catalase negative and they would not. So once again, if you needed to identify bacteria, very often bacteria can use, some bacteria can use one sugar as a metabolite and break it down, while a different type of bacteria might not. And so if you add bacteria to a sugary sample, they might be able to digest it, which would change the color of the pH indicator, turning it from pink to yellow, or maybe even accumulate a gas bubble. And this could be used then to identify the bacteria. Some bacteria need oxygen and will only grow at the surface of agar. Some can't live with oxygen and will only grow in deeper parts. Some are motile because of their flagella, and so therefore would be able to spread throughout a deep of agar, a tube that you stabbed with a probe to introduce bacteria there. And so if you need to identify bacteria in a patient or in an ecological sample, there are a number of microbiological tests which do that. And so I have a playlist which runs through some of the common ones, and a lot of biology students end up taking microbiology classes, where very often the last half of the course is you're given an unknown bacteria that you then have to identify. I then go through a few other techniques, so here's a technique where you can make a series of dilutions of a bacterial sample and then plate them, and then figure out how many bacteria were in the original sample. So once again, like a lot of times, a pool or a public beach will be closed if there's, this many coliform bacteria per, 100 milliliters. Well, how would you count that? Well, let's say you can make this dilution series where you keep taking, say, a milliliter from this, you put it into this sample, mix it, then you take a milliliter of this one. You'll notice that the pink gets more and more diluted as you go along. And then when you plate them, all right, so you make a series of plates. Let's say you had a tube which had diluted the bacteria by a factor of 1,000,000. So you had a sample from, the river where people were swimming, for example, and you diluted by a factor of 1,000,000. And then on the plate, you count, say, 5 bacteria, etc. And you ask how many were in the original. Well, if you diluted it by a factor of 1,000,000, then you would multiply the number 5 or whatever by 1,000,000. So there were 5,000,000 bacteria per milliliter of the original sample. Okay, so these 2 videos that I made were simply providing an overview of the playlist of videos covering diverse aspects of prokaryotes. And, obviously I wanted to end this right now, but, just a quick comment. Bacteria, they're the most common organisms on the planet in terms of number. And even then, just as far as mass, bacteria far, far outweigh all of the animals combined. They certainly live in so many diverse environments that when we think about where life exists on our planet, if we're excluding like all of the deep sediments underneath the bottom of the ocean, all of the deep rocks, all of these extreme environments, you're missing, upwards towards half of all of life on Earth. These were the first living things, having the planet to themselves for about 1.5 billion years, a third of the history of life on Earth. And much of what evolved in these early bacteria are what the more complex form of life, like ourselves, then utilize to be more complex. They are essential in food chains, all right, providing organic carbon, organic nitrogen, which then other organisms rely on, of being decomposers, which recycle nutrients of things which have died. And then finally, as we'll see in the next topic of protists, the eukaryotic cells, like the ones that make me, they have little things inside them called mitochondria, all right? Mitochondria seem to be the descendants of bacteria that learn to live inside bigger cells. And so not only do I have, say, 2 pounds of bacteria associated with my body, more cells than I actually have human cells, my human cells actually have the descendants of bacteria in them. And so if you're a biologist, prokaryotes are important in your understanding of biology in so many ways.